Okay. Hello, everybody. Thank you so much for listening to New Technology for Advancing Applications of Multimodal Imaging. My name is Giorgio Bonmassar. I'm an associate professor in radiology at Massachusetts General Hospital, Harvard Medical School in Boston. This presentation will be bestly followed if you also looked at or clicked at the three previous presentations from Dora Hermes, Catie Chang, and Laura Lewis. So multimodal imaging is quite popular, and the reason why it is used quite often is that there is no single modality, imaging modality that allows us to have both very high temporal and spatial resolution. So today, I'm going to focus mostly on the combination of multimodal combination of EEG and fMRI. So we are interested both in recording invasively, electrophysiology, and non-invasively. So obviously, the most common and easiest form is the non-invasive, which is EEG or electroencephalography. And so, but in practice, in reality, we really want to know what are the activity of the underlying neuronal cells in terms of postsynaptic potentials. So when are these potentials really visible in the EEG? Well, it depends really on the populational size. People is kind of basic rule of thumb is that a patch of cortex of at least 5 millimeter by 5 millimeter has to be activated in order to be seen, and also not all the neurons are visible. Like for instance, stellate cells activity is very hard to see. The other thing is we heard from Dora's presentation is that we really like to, we can only pick up signals that are highly synchronous with each other. Also, as the population is deeper and deeper in the brain, like in the brain stem, we have to use special tricks like averaging a very large number of brainstem evoked potential in order to measure that. The orientation is important for capturing or not capturing neural activity. And there's also a possibility of cancellation or shielding, in which you have two activities that are opposite in two close by cell side. And of course, the SNR's issue is always in order to capture very small signals. Well, in general, we're going to talk about non-invasive, as I said, and mostly we'll be only talking about high-density EEG. And then when we talk about the invasive, we will talk both about deep brain stimulation and ECOG, which are subpleural electrodes. So the high-density EEG net are quite used quite a bit, widely used in neuroscience because of the very high spatial resolution that can be acquired through EEG. But we wonder what are the effects in terms of safety and MRI quality in very high fields. Basically, these electrodes are very packed together. The electrodes contain a lot of metal, and they're connected to the amplifier through wires. So the metal in the electrodes and in the wires can produce susceptibility artifacts on the MRI, especially in high fields, and can produce RF and homogeneities. And the very, very long leads can pick up the RF, which is used in the MR to elicit signal from the tissue. And this can be kind of dissipated back into the electrode, and they can produce some heating. we're going to talk about this effect called antenna effect quite a bit along this presentation. So in terms of safety, the United States Food and Drug Administration often talks about the specific absorption rate calculation. And the specific absorption rate is this formula here, which is the sigma C, which is the conductivity of the tissue, point by point, divided by 2 times the mass density, point by point, and multiplied by the magnitude of the electric field, again, point by point. So usually the standard is to reduce the overall SAR average of the entire head to less than 3.2 watts per kilogram. And this quantity is more or less equivalent to not raising the temperature of the head for more than about 1 Celsius, which is kind of a natural thing that may occur as you expose your head, essentially, like, for instance, in the beach for 1 hour. How do you compute this SAR? Well, you start with the head model, and this is a head model that we developed and has a very, very high anatomical accuracy in a large number of tissues. Each of the tissue, of course, have conductivity, so we have assigned conductivities to each tissue according to that basis. And we talked about the antenna effect, and the antenna effect occurs because copper is extremely conductive, and so it's a very, very good antenna. So obviously, if we reduce the conductivity of the leads, then we have an effect both on the electric field, the induced currents inside the leads themselves, and the specific absorption rate. So many manufacturers of EEG caps and nets used to put a 10K resistor on top of the electrode. However, we and others have shown that this does not change almost at all the local SAR. And basically, the presence of all this metal still produces like a shielding effect. So it will change both the SAR and the electric field despite the presence of the electrons. So the solution that we provide is what it's called the InkNet, in which we use traces that are printed with the conductive ink, in which we can control the conductivity and the electric properties of the traces. And this is a high density 256 electrocap. This has been designed through numerical simulations, in which we use our head model. And we attach the InkNet on top of the head model. The head model includes also the shoulders, because the subject is placed into an MRI. And this is a birdcage coil. And the numerical simulations allowed us to compute the peak SAR and to estimate the heating of different cases. So the numerical simulations allowed us to study and predict the peak SAR and the heating with different conductivities of the traces. So as we pick up, during manufacturing, we picked up conductivity that it's both safe and produces EEG with high quality. And so we tested this at 7 Tesla. So the testing was done using head phantom with the same electric conductivities and permittivity as white matter. And we inserted a number of thermal probes, which were very close in contact to the electrode themselves. Since the probes are connected through plastic fibers, we could only put small number, a certain number of sensors, because otherwise the entire phantom would become a plastic. So we decided to subsample. So since we have all these electrodes, we decided to choose to monitor the electrodes that, according to simulations, had the highest possibility of heating. And also we based the selection of the electrode based on the B1 field, which is the peak of the RF fields. And so when we did the measurements, we compared a case of no electrodes with just a phantom and the inknet, and we saw that in practice there is no heating at all. Then we looked at the quality of the MRI images, and we did this following experiments. We computed the eccentricity map. So in the 1970s, Eric Schwartz showed that a ring in the visual field, if you were saccating, if you were kind of fixating right in the center of the field, would be represented as a line into your cortex. And so this is called the eccentricity map. You can show like how a small area of the field on the visual field is projected into cortex. And so this is how a normal eccentricity field looks, and a very small portion of about 2 degrees of the visual field corresponds to a large area in the cortex E1. When we compare the eccentricity maps with the MRI only, we see that typical eccentricity map, when we have the subject wear an inknet, basically the eccentricity map is almost unaffected. However, when we use a copper-based net, commercial net, we see that we lose most of the eccentricity map information. We are now working on a version for a child called the NeoNet, and this actually is based on thin film technology. I'm hoping to present this next year at OHBM, and all our safety studies and the design of the inknet itself is based on a 20-month-old model that we have developed called Martin, and has more than 90 tissue. And this time, instead of just working on a head model, we have developed the entire body model, because the child is so small that the entire body is basically affected or influenced by the B1 field or the RF generated by the scanner. Okay, so this is the transition slide between non-invasive EEG and the invasive recordings. And so just a joke, the bad news is that we left the clamp in your temporal lobe. The good news is that you won't remember what I just said. And so we're going to now talk a little about some of the applications of chronic implantation with electrical stimulation. So the electrical stimulation is used quite often to treat neurological disorders, and deep brain stimulation is FDA approved for their use in dystonia, Parkinson patients, essential tremor, and obsessive compulsive disorder therapy. When you introduce neurofeedback, you also have the ability to control epileptic seizures, cochlear implant for restoring auditory function, and spinal cord stimulators for pain. We're going to focus on deep brain stimulation, and the deep brain stimulator is made of an electrode which is placed on different targets, including different targets in the basal ganglia, including STM, GPI, and VIM. And then this electrode is connected through long wires all the way to stimulator, which is called the IPG, implantable pulse generator, which is very much like cardiac defibrillator. And, and these long wires are really the ones that, again, can be affected by the antenna effect. And so, unfortunately, in the Cleveland Clinic in 2005, we recorded an accident. So, a patient undergoing MRI for back pain, so completely unrelated with movement disorder, had a bilateral DBS implant with different lengths. And upon removal from the MR, the patient developed right hemiparesis. So we study this case because basically the MRI is considered a primary tool choice for clinical diagnostics. And there's a large number, many millions of recordings done each year for clinical and research. And there are 500,000 patients over the world with implants, and often the MRI examination is denied because of compatibilities. So we used our head model, and we implanted the electrodes and looked at the resulting electric field. And you can see in this plot, the electric field kind of really spikes up compared to the case of no implants at all. This corresponds to very large changes in the specific absorption rate. And we don't look anymore at the average because the size where the peak is very, very small. But we look at a different standard, which is the IC 10 grams. And in this case, the IC 10 grams, in the case of DBS, was shooting up very, very high compared to the no DBS case. And so this is why essentially the subject had a hemorrhage, because the tissue around the electrode heated up very much due to the large amount of currents that were injected through the electrodes and were picked up by the long DBS leads. Well, if we follow the same approach that we did for the InkNet, we could just say, okay, let's change the conductivity. Since copper is too conductive, we can just go down. And so then we can bring down the 10 gram SAR to a safety level. And this will definitely happen. And this is confirmed by the simulation. However, the amount of current that we have to inject into the wires and electrodes when they become not very conductive is very large, and that will quickly discharge the battery of the implant, which is not desirable. So we had to come up with a more clever idea, more sophisticated idea, and we worked on what is called the RTS theory. And so basically, we worked on material made of two different materials with two different dielectric property, sigma 1 and sigma 2. So the idea here was to make the wire induce currents much smaller than the currents they would had otherwise. And so the idea was to design a very bad antenna. The other part of the design is that we wanted to amplify or maximize the type of reflections. So the reflections would reflect back the signal towards the IPG or the neurostimulator and away from the electrode where the burn or the lesion occurred. And design was done using what is called ASTM phantom, which is the standard phantom used in the safety measurements. So we basically design the sigma 1 and sigma 2 in order to have the minimum 10 gram average and still maintain pretty good overall resistivity of the wire, about 400 ohms. Usually a wire is about 100 ohms for the traditional implant. So this new design allows us to reduce the peak of the SAR, 10 grams, the peak SAR, and also to bring back the peak of temperature that we would otherwise have with traditional platinum iridium wires down to the case of no lead at all. So this was tested into an MRI at 3 Tesla, and we saw that the standard 3389 Medtronic implant heated up about 10 degrees, while our implant stayed just about Celsius above baseline. And the discharge of the battery inside the IPG or impulse implantable pulse generator basically stayed the same with respect to the normal implant. Same thing when we did the simulations with a realistic head model, we saw the same kind of behavior with in terms of SAR peak. We finally made a version, implantable version, using the same diameter as a platinum iridium wire that is used in implants. And you can see here the images in the optical microscope and in the electron microscope. Here you can see the changes in conductivity over space. Well, I'd like to talk about another type of implant, which is the cortical implant. And these are usually used for neurosurgery, for epilepsy, pre-surgical planning. This is the effect of a grid, standard grid, commercial standard grid made of metals. And the metal is clearly visible, and the effect of the very heavy and very rigid grids are visible in the MR as well. Even more visible is the effect on a CT, and this occurs because the CT material is made because the implant material is made of a metal which is extremely dense and blocks the penetration of the X-ray of the CT, producing this kind of artifact, which makes these images completely unusable for radiology. So we came up with a new type of solution based on absorbable gelatin film, which is used currently in neurosurgery. And we used, also in this case, conductive inks. The design was done, again, using numerical simulations, using what is called FTTD. And the numerical simulations were, as usual, validated with measurements with the MRI. These are flip angle images of B1. And so the phantom only and the commercial electrodes are visible in terms of artifact, while the PTFOS, which are our electrodes, the artifact disappears. So we implanted the PTFOS in mice, and we looked at images of scanning electron microscope before and after, and we saw that the electrodes were essentially intact, in both in terms of micro images. Then we compare the case of the gel film, which is the control, just the biomaterial used in surgery since many decades. The conventional platinum grade, or the conventional commercial grade, and now PTFOS. And so this is the gel film only on the first column. The second column is the commercial grade, and this is our PTFOS. So the H&E stain showed the general structure of the tissue, and the control is very similar to the PTFOS, while the standard images show perturbance in the general structure of the tissue. We did fluorogade staining to look at dead or degenerated neurons, and we saw that the gel film again brought it back to the PTFOS, while we saw dead neurons in the case of the standard electrode. We used Nuon for the stains of neuronal nuclei. The nuclei density stayed the same for the PTFOS and the gel film, while the standard, there was a reduction in the nuclei. We used IBA1 to look at microglia, and we didn't see any microglia, which is like inflammation response of the body, while the stains of microglia were present in the case of the standard electrodes. And finally, we looked at the Bielchowski silver stain, which looks at nerve fibers, and the nerve fibers look healthy both for the gel film and the PTFOS, while there was disruption in the case of the standard electrodes. We also did imaging tests of standard, low-grade, and PTOF, and the CT is completely clear in terms, is completely invisible in the case of PTFOS, while the standard produces a large artifact. Same thing for the T1 images, the T2 weighted images, the flare, and 7 Tesla flash. The standard produces a very, very large artifact, while the PTFOS and the no-grade produce no artifacts at all. So in conclusion, we saw non-invasive, talked about non-invasive technologies, mostly high-density EEG, and we provided some new materials that could be useful for future implants that are both safe and with high MRI quality. And here I like to conclude with a set of future readings if any of the viewers are interested in this topic.
Hi, my name is Sepideh, and I'm part of the educational course this year, and it's all virtual, but I hope it will be informative to you. I will be talking about connectivity, large-scale functional connectivity in electrophysiological modalities with a focus on EEG, recorded concurrently with fMRI, and some data from invasive recordings, surface cortical recordings, or ECOG. I hope to convince you in this talk that it's very informative to actually estimate functional connectivity from fast oscillations, even if you have surface data and need to source localize them. So let's dive right in. This is an overview of one possible pipeline that you would use if you have surface-based EEG or MEG data. You would start with the pre-processed signal here to the left. And then typically, you might want to break it down into different oscillation bands, although some studies use very broadband signals without filtering them. If you want to break your signal down into different oscillations, you have the option to just go with the canonical frequency bands, or you can use more elaborate measures, for example, individualized estimations of the frequencies depending on the power spectrum, as Dora has been showing in her talk, which I hope you've seen. Then you will take the band-filtered signal or the broadband signal and source localize it to your cortical surface. And then you will average that signal or localize that signal to an atlas, to a whole brain parcellation. Now, note that this is a different approach than what source localization typically was developed for, which is thinking about a very limited number of task-related sources in the brain. Here, we're talking about a whole brain parcellation and source localizing oscillatory activity to that entire atlas. You will then, for every parcel, have an ongoing signal, a signal time course, for which you can estimate, for example, the power envelope. That's the blue envelopes shown here for each of the frequency bands. You can also estimate the phase of these frequencies, and then depending on whether you're interested in the amplitude or phase, you can estimate functional connectivity as pairwise, that means between region pairs, pairwise correlations of the amplitude envelopes, or as phase coupling, such as measures of coherence or phase locking. The end result of that is a functional connectome, a whole brain functional connectivity matrix for each one of the frequency bands, similar to what you might be familiar with in fMRI. When we do functional connectivity in electrophysiological data, however, we have a much richer signal than with fMRI, and that brings about multiple questions of how you should go about estimating your connectivity. When we look at the hemodynamic signal, we have just a pre-processed ongoing signal that we use, for example, for Pearson's correlations. When it comes to electrophysiology, again, as I mentioned, we have the option of conceptualizing functional connectivity as the coupling of amplitudes across our brain regions, or as the coupling of phase, or even higher order cross-frequency measures, such as phase amplitude coupling. The other complication or richness of this data is that we are actually dealing with 4 orders of temporal magnitude. The signal goes all the way from infraslow, which is the typical range that we use in fMRI, to the high level of the canonical oscillations in the gamma band. So this is another point that makes the data much more complex than what we know from fMRI. The outline here is at the same time really the conclusion of my talk. So I'll start with that. I will be emphasizing the stability of intrinsic functional connectivity in brain oscillations. By intrinsic, here I mean functional connectivity that is not driven directly by cognitive engagement or external tasks. So it's not the change in connectivity due to external task demands, which is what EEG and MEG are typically used for. This is really ongoing intrinsic functional connectivity. Can be measured in resting state, but can also be measured in other mental states, as I will point out at the end of the talk. I'm emphasizing the stability of this functional connectivity, both across space and across time. I will show that stability in comparison to concurrently recorded hemodynamic signals. I will also show you data that emphasizes that the spatial organization of functional connectivity of oscillatory signals is actually very similar between phase coupling and amplitude coupling. And finally, I will show you the stability across mental states to show that functional connectivity really is similar whether or not the subject is cognitively engaged or during resting state. And this is really conclusions that we know from fMRI and now have recently found is extending to fast oscillations as we see them in EEG and MEG. So let's start with the stability, the spatial stability of static connectivity. So here is one of the earlier papers that was really demonstrating that you can generate the canonical neurocognitive networks that we know from resting state fMRI, that you can generate them in resting state MEG as well in whole brain source localized data. This is seed-based connectivity, in this case in 1 through 150 hertz broadband signal, estimated as amplitude coupling. Another thing that you can do is starting off with your channel-wise data, either EEG or MEG, and then generate the whole brain connectome. So similar to what you do in fMRI, where you average the fMRI signal to the different parcels of the atlas and then generate your connectivity matrix, for example, using Pearson's correlations, you can do something similar. Here in EEG, you would source localize your data to the same atlas if you have concurrent data and are interested to cross-relate these two data modalities. And then from there, you estimate your functional connectivity measure. So this is just an example. You can use coherence or imaginary coherence. And then you will get one connectivity matrix per frequency band. So here, this is very similar to the outline that I showed before to the processing pipeline, except that I'm here now emphasizing that you can do it for concurrent data in the same spatial space so that you can directly compare this data. And this is one of the studies that did so, a direct connection-wise comparison of functional connectivity in EEG on the y-axis and in fMRI on the x-axis. So every dot in this plot is the connectivity value for one connection, meaning for one region pair. This is averaged across all subjects. You see the functional connectivity strength edgewise for the beta band in this case as a function of fMRI connectivity strength. You see a significant moderate size correlation. Interestingly, a very similar picture has been shown for amplitude coupling. So it really doesn't seem to matter whether you're looking at phase measures or amplitude measures. They both are strongly related to concurrently recorded fMRI connectivity in spatial terms. Some of you may be aware of the literature of the mid-2000s where the reliability of resting state functional connectivity from fMRI was demonstrated because of its correlation to structural connectivity as estimated from DTI. Something very similar has been done for EEG. So here in the top plot, again, you see the EEG function connectivity strengths for the beta band here on the y-axis. This is as a function of structural connectivity on the x-axis. And the correlation is negative here simply because of the type of structural measure that was used. But important is the magnitude of correlation. It's a very substantial relationship between the edgewise strength of beta band coherence and the edgewise strength of DTI estimated structural connectivity. And in this particular study by Versich et al., this connectivity of EEG to structure was actually stronger than the relationship of fMRI connectivity to structure, shown here in the bottom plot. So this was really convincing for me personally, that it makes sense sufficiently, in spite of all the technical problems and in spite of the issues with the inverse problem and source localization, that it is possible and feasible and informative to use scalp recordings for whole brain connectivity. I don't want to really go into detail of this study, but I felt I had to mention that there are multiple studies out there that go beyond cross-modal correlations. So instead of just correlating EEG with fMRI, for example, you can go ahead and do bimodal decomposition of the data together. Here we're using a method developed by Enrico Amico and Joaquin Gonier. We applied it then to concurrent EEG and fMRI with them. So very briefly, here you see a connectivity matrix, Pearson's correlations for each individual subject in fMRI. And then we estimated connectivity for every frequency band of the concurrently recorded EEG, again for every subject. We vectorize these connectivity values and then just essentially put them together, the EEG and the fMRI, and do a decomposition of both together to find stable components that co-occur between EEG and fMRI over subjects. The difference of this decomposition to many prior decompositions of EEG fMRI is that the values that enter the independent component analysis are actually the connectivity values, not the time courses. Again, I don't want to go into the details, but what we learned from this study is that there is a stable component that has both an EEG part and an fMRI part, and both of those parts spatially reflect intrinsic connectivity networks, the typical neurocognitive networks that we know. So really again emphasizing that those networks exist in static EEG connectivity as well. And so all of what I've shown right now is related to functional connectivity as it's averaged across the entire recording. But many of you are aware of the discussion of time-varying dynamics in connectivity, so how functional connectivity changes over time. And what I'm going to show next is that even those changes, the temporal changes of connectivity, are related between fMRI and concurrently recorded EEG. So this is one particular study, one example, where we estimated sliding window connectivity in both modalities. Sliding windows are one typical way, probably the most commonly applied way in fMRI literature to estimate functional connectivity changes over time. So we estimated Pearson's correlations every 1 minute in a sliding window. So the window is sliding every 2 seconds, but the window is 1 minute wide. We did the same for coherence in EEG for every given frequency band. So this is an example for one frequency band. And then we estimated for each connection, so each point of this connectivity matrix, we estimated the mutual information over time between the EEG and fMRI. We compare then that mutual information estimated here to mutual information from an appropriate null model. And then we just do a statistical comparison. What we found was actually really surprising to us, namely that in each of the canonical oscillation frequency, theta, delta, theta, alpha, beta, gamma, no matter what frequency you're looking at, the EEG and fMRI sliding window connectivity strength is significantly related to each other, has significantly higher mutual information than the null model in almost every connection of our connectivity matrix. So again, for every frequency band and for every connection, any change in connectivity strength, any increase or decrease is coupled with a change in functional connectivity in fMRI. So this is in resting state data. And I'm here just showing the top 200 connections with the strongest tie between EEG and fMRI. The point I want to draw from this figure is simply that regardless of what EEG frequency you're using, the relationship between EEG and fMRI is spatially very, very distributed. Further analyses showed subtle differences for sure between the different frequencies, but it is not the case, for example, that alpha band connectivity would only occur in occipitoparietal areas and co-occur with fMRI there. It's really distributed everywhere. This is the example for alpha. It holds true for all frequencies. Now I'm coming to the stability across the different coupling measures of electrophysiological data. So I'm showing phase versus amplitude coupling. And here I just wanted to mention that all the work that I'm showing after this, I've decided to show you from a publicly available library. So this might be a great resource for all of you who don't have direct access to this precious invasively recorded human data. Electrocorticography data. It's a public repository. It's further described in this 2019 paper of Kai Miller. This link is in that paper. It takes you right to this webpage. No overhead. You can directly go in and download these the data of the patients from these different tasks. So everything I'm going to talk about is something you can go ahead and try at home. So again. Big thanks to Kai and his colleagues for sharing this data. So moving forward, we have a much more reliable electrophysiological measure because we're no longer recording from the surface of the brain, no longer recording from the surface of the scalp, but we're directly recording with the electrodes that are lying on the cortical surface. So we have no need for source localization and can really get at the core of a lot of the questions without the worries of surface data. So this cartoon illustrates what I mentioned at the beginning of the talk, namely that functional connectivity can be conceptualized as phase coupling, for example, between regions A and B, So the two regional time courses are shown here in the two colors, and the phases are pretty well coupled to each other. From a biological point of view, you can, however, define functional connectivity quite differently between regions A and C. This would be the amplitude of those signals being coupled together. So the amplitude of the green and the blue time course are changing together, but not the phase. And alternatively, you can have connections like C and D, where both phase and amplitude are connected to each other. And we don't know in the real world whether this version of co-occurrence of phase coupling and amplitude coupling really holds true or not. So I hope that the following, what I'm going to show you, is going to help you with future decisions if you are using electrophysiological data of where and when you should use one or the other coupling measure, or at least emphasize that if you do static connectivity, they will be very similar to each other. So here to the left, I'm showing 4 example subjects of the full subject data set. They were engaged in a simple motor task here. And to the left, I'm showing functional connectivity during that motor task. This is finger movements for amplitude coupling. And to the right, I'm showing the connectivity matrices for phase coupling. Now, remember that we don't have whole brain coverage, so the electrodes are shown in black dots here on the brains. So these matrices cannot be directly compared across subjects. Each one of them has their individual electrode coverage. So this is widely distributed connectivity, but not whole brain. So typically running between frontal and temporal lobes in these patients. What you can visually appreciate, though, is the similarity between the left and the right matrices, meaning those that are estimated as coupling of amplitude and those that are estimated as coupling of phase, and in this case, using phase blocking values. To quantify that, I'm showing the spatial correlation between the amplitude coupling matrices and the phase coupling matrices for 18 different subjects. So this is the cross-measure spatial correlation between phase coupling and amplitude coupling. The black dots are each one subject, and these are the different frequency bands on the x-axis. You can see that there was moderate correlation in every given band between the spatial organization of phase coupling and amplitude coupling. And let me actually show you the legend here. The rest, what we're showing, that we're showing in this figure, is that the gray line is the correlation, the spatial correlation between phase coupling and amplitude coupling during the pre-stimulus time. So before the subjects were actually moving their fingers, they're not doing anything. This is totally intrinsic. And you see that the correlations are almost identical. So if you subtract one line from the other, you are left with the data here in orange, the orange data points. And what that emphasizes is that the type of functional connectivity that is highly correlated between phase coupling and amplitude coupling is actually intrinsic. It is not related directly to the task. It's driven in an ongoing manner. We have also looked at this correlation at resting state, and we find, again, very, very similar relationships between phase and amplitude coupling at resting state. Without showing you the details of the data, just to save us some time, I'm just giving you some further conclusions from additional analyses that we did. The task-related phase and amplitude coupling changes relative to pre-trial baseline are not spatially related between phase coupling and amplitude coupling. So again, the connectivity matrices estimated during task and during baseline, they both show a cross-measure relationship between phase coupling and amplitude coupling. But if you're looking at how the functional connectivity changes from baseline to task, okay, so baseline corrected task-related functional connectivity is not overlapping spatially between phase coupling and amplitude coupling. So they do have different functions. These two coupling modes have different task-related functions. Just in intrinsic terms, in terms of their ongoing spatial organization, they are tied with each other. A similar conclusion can be drawn when looking at the time-varying connectivity dynamics in a task-free setting. So, for example, if you would take the task data or you would take the resting state data, and you would see how over time in a sliding window procedure, these fast oscillations are changing their coupling in phase coupling and in terms of amplitude coupling. Those changes in functional connectivity over time are not at all correlated between the two connectivity measures. So phase coupling and amplitude coupling are completely decoupled from each other, are unrelated when it comes to their changes over time. So they're not identical. Remember, I'm not saying you can just use one measure and forget about the other because they are going to show you the same thing. They're actually quite different. They're different over time, and they're different in terms of how they change from baseline to task. But in terms of intrinsic connectivity, that means that the connectivity that is not related to the task-related changes, they're extremely spatially similar. And that conclusion is reiterated here. All right, so I'll let you digest that for a moment. And then finally, I'm going to get to my final message, which is that both phase coupling and amplitude coupling of fast oscillations are extremely stable, independent of what cognitive setting the subject is in, whether they're cognitively engaged in a task or whether they're at resting state or whether they're in a, various different kinds of tasks. So to illustrate that, I'll start off with what we know from the fMRI literature. So here to the left, you see work from 2014. Two different labs concurrently showed that the spatial connectivity organization during tasks and during resting state here to the right are spatially extremely similar to each other. In fact, this particular paper by Mike Cole estimated the spatial correlation at 0.9 between the connectivity profile during tasks and during resting state. A more recent paper by Katerina Graton actually shows a comparison of magnitude of this stability within subjects over different tasks. So the different tasks are color-coded here. At the bottom, you have the legend here. So the different colors on the axes are different tasks, including resting state. And then these are different subjects for which essentially this spatial relationship of the connectivity matrices has been quantified as z-scores. And what you can see is that there is a very strong spatial similarity of the functional connectivity profile within each of the subjects, irrespective of the cognitive content, whereas they are pretty discordant across subjects. So again, this is just summarizing that the fMRI literature has shown us that functional connectivity is showing only very subtle changes that are related to our cognitive and mental life. Most of the functional connectivity is intrinsic, is independent of what's going on in the outside world and the type of task that we're engaged in. Long story short, we found something extremely similar, again, in the same surface-based electrocorticography data that is publicly shared. So here to the left, I'm showing phase coupling. And the different colors on the axes are showing the different kind of cognitive tasks. So we have fixation, which is basically eyes open resting state. And we have a motor task, we have a highly cognitively engaging end-back task. So these are very different kinds of tasks. And then we have the pre-stimulus baseline of the motor task. So across these different mental conditions, you see the value of spatial correlation within each subject. This is average values across all subjects, but the correlation between the different mental states was calculated within each subject. You see that they're extremely high values. So in the upper triangle, you see the color coding. This cyan green range is already moderately strong, and it goes all the way up to very strong correlations. So the connectivity matrix, the static connectivity matrix, is very spatially similar across different cognitive states. This holds true for phase coupling, and we observe something very similar for amplitude coupling. You see that the value of the correlations is overall lower, but they are all significant above the permutation null model that we're demonstrating here in the bottom triangle. So less strong correlations, but still highly significant for amplitude coupling as well in terms of the similarity of the spatial function connectivity organization and oscillatory in oscillatory terms. And so with that, I will leave you again. You can go back to the outline slide, which basically has all the core conclusions of this talk. I hope that I've convinced you that it does make sense to branch out into electrophysiological measures, use EEG, use surface-based EEG, surface-based MEG, or intracranial electrophysiological data, and look at the rich and fast range of brain activity instead of focusing solely on hemodynamics, which is a little more represented in our society at the moment. So I hope I convinced you that it is methodologically possible and that it is extremely informative to look at wide scale functional connectivity, both during task and during resting state. So we have summarized these conclusions in this recent review paper. You find a lot of references. It has a very long reference list. So hopefully it will lead you to where you need to go if you want to dive into any one of these points in further detail. And I'll leave you with thanking my lab members, especially Jonathan and Parham, who did most of this work, as well as those who very graciously shared their data with us. Thank you.
Hi. Today I'll be talking about ways to combine multimodal data offline when simultaneous or invasive methods aren't available. I'll be focusing on fMRI and MEG, but the principle we're talking about applies to fMRI and EEG and potentially other modalities as well. Okay, so I will first outline some of the basic challenges, which are clear if you've seen any of the other talks in this session. I will then outline representational similarity analysis, which is the framework that we'll be using to combine the data. And I'll give a basic outline of these steps by way of going through an example from some recent work that we've just submitted. And I'll show some further examples to illustrate extensions to the basic technique. So you get a full range of the space that you can operate in. And finally, I'll summarize some caveats and future potentials of this method. So, we all know that the brain is a spatio-temporally dynamic system, right? It happens over time and space. And the trouble, of course, is that most of the methods available to us are pretty good at one or the other dimension of this kind of activity, but not both. And as we've seen, there are invasive methods like ECOG or simultaneous EEG fMRI setups, and they carry some of their own sort of clinical and technical constraints, but also they're just not available to the vast majority of cognitive neuroscience researchers out there. And finally, we haven't fully modeled the brain, right? We don't have explicit neuronal or hemodynamic models for arbitrary brain responses. And so we look for some other ways in which we can represent sort of population level neural activity. And so the way that we address all these challenges in this case is representational similarity analysis. Now, this concept has been around a long time. I'm showing a slide from Nico Kriegeskorte's review in 2013 to go through some of the basic principles and intuitions. So you can see here that a stimulus evokes, multivariate activity, right? So that is the first main point. Whether it's a hand or an umbrella, it evokes activity in a pattern in a set of voxels or neurons, or like a deflection, a set of deflections in MEG or EEG sensors, right? And we can use that multivariate response to compute an abstract dissimilarity space. In other words, we can abstract the sensor space into something more normalized. And the similarity space is embodied in what we'll call the RDM, the representational dissimilarity matrix. Okay, and that is just the combined pairwise dissimilarity measures of every pair of stimuli. And so you can see it's a square matrix, the size of your stimulus set. The diagonal is undefined, and it's symmetric about the diagonal, so you can pay attention to just one or the other triangular half. So now matrices are very versatile, right? So RDMs have the nice property that they're directly comparable across different ways of measuring them, unlike the raw signal source space. So in fMRI MEG fusion, we also have this intuition of isomorphism, meaning that similarity structures for a given representation should be similar across MEG and fMRI. Okay, so going through each of these basic principles in a little bit more detail. So you want a representational space that covers the effects and representations that you're interested in. And you want to do this by having a large and rich condition set. Now the classic examples of this are big hierarchically categorized images, image stimulus sets, but there are other modalities available too. So in some ongoing work, we've been presenting Braille tactile alphabetic letters to blind readers and the visual print equivalent to sighted controls. Jody Cullum's group has presented 6 physical objects to subjects in the scanner, but each of them was subject to 3 size manipulations and 4 potential interaction options, and that made for 72 total conditions. And the example that I'll go through with more detail now is the naturalistic. There's a set of 80 naturalistic sounds grouped into 4 sort of high-level semantic conditions. And in this case, participants just listened attentively but passively to those sounds. So the analysis, generating these RDMs that we're so interested in, first you do the analyses independently for the separate modalities, right? And so in our case, that would be separate sessions. So you do an MEG decoding analysis. In this case, you decode the multivariate patterns of the whole brain signal at every time point between every pair of stimuli. And what that means essentially is that you generate a whole brain RDM across the entire stimulus space for every time point. In this case, for a 200 millisecond baseline and 3000 millisecond epoch, that gives you about 3200 RDMs. Now, in this case, we used SVM linear classifier accuracy as our dissimilarity measure. So highly dissimilar stimuli would be very highly classifiable, accurately classified. And so given that an RDM is just a collection of pairwise classification accuracies, you can collapse those, right, and just create an overall classification accuracy time course. And so a lot of temporal decoding papers that you've seen in the last 6-7 years have this kind of results. But we don't want to throw away our pairwise approach. We don't want to throw away the similarity structure. We hold on to that. So then we do the same thing in fMRI, the difference being that we are not iterating the whole brain over time. We are iterating parts of the brain over space. And of course, you can do this with a hypothesis-driven ROI approach, but the most spatially unbiased form would be just a single voxel at the center of a searchlight volume, and you iterate that across every voxel in the brain. So instead of time, you get RDMs over space. So the fusion itself is very straightforward. You've converted everything into RDM space, similarity space, and by correlating the MEG whole brain RDM at each time point with all the RDMs across the fMRI volume, you generate a series of correlation maps. And in this visualization, they're like frames of a movie, and they show where and when the MEG and fMRI pattern responses are similar. So here's another sort of overview to recap the whole process. You have your experimental conditions, each of which generates a distribution of responses in their respective native signal spaces. And then you convert that to a representational similarity space across regions or time. And finally, you fuse these representations in the RSA framework to generate either the movie frames like I was talking about, or equivalently, you can visualize this as a set of time courses by region. So let's look at the movie visualization first. This is from our auditory data set. And you can just, you can see this thresholded movie shows the expected early superior temporal responses to our set of naturalistic sounds. And they propagate rapidly, sort of anteriorly, superiorly, and posteriorly out beyond the temporal cortex and beyond classical sort of auditory cortex. Now, what's interesting to note and important to bear in mind is that this is a map of correlations across time, right? This is not raw deflections. It's not a set of scalp topographies like in MEG or EEG. So it's a higher level representation of where the MEG and fMRI signals are most similar. Okay, now this is very interesting and unbiased, but you may be wanting to test a more specific hypothesis about specific ROIs. So we can also look at this data in a more targeted way. And here you can see that you can parcellate the fusion response with a time course for each of a discrete number of ROIs. And so here you see these time courses color coded to the corresponding ROIs. And the bars here indicate temporal regimes of significant correlation. So not only do you get this peak and onset information, right, you can also see the temporal dynamics of each region, how transient, how sustained, whether there may be some recurrency and that sort of thing. So it really lets you sort of pick apart the spatiotemporal dynamics in much finer detail. Okay, so now if let's go through some other examples. And by way of showing the first example, the original, the canonical application of this method is with the ventral visual stream of object processing. And I'm kind of citing two studies here from Radoslav Cichy. And so here you can see the familiar early to more anterior propagation of visual responses. And here you see the two ROI time courses, just V1 and IT, and they sort of corroborate this idea that the signals, the MEG and fMRI signals, correlate early in V1 and later in IT. Okay, so that's well and good, but one other thing you can do with the RSA format is that, remember, you have a large condition set, right? And the condition set spans a range of categories. However well you curate it, you can interrogate that structure. And the way that's operationalized here in this little figure is that you don't just have this agnostic RDM for your searchlight. You have something that, in this case, distinguishes between half the stimuli, which were animate objects, and half that were inanimate. So if you're interested in just animacy discrimination, then you can operationalize that as this model RDM. And similarly, you can operationalize, other subcategorical distinctions. And that means you can sort of partition this movie and the data underlying it as these more semantically specific, content selective visualizations. So you can use this method not just to show responses generically, but content specificity. So here's another example beyond just the feed-forward processing suite. What happens if you present a stimulus very, very briefly, right? So in this example from Yalda Mosenzadeh and colleagues, I've truncated this a bit to start the visualization at about 60 milliseconds. So you can see this is similar to the previous slide, right? But the presentation was so brief, you can see the early response die off, and then it comes back, right? So you're not just seeing a normal feed-forward sweep. Here, the authors truncated the feed-forward sweep so that they could reveal, unmask some feedback here from higher level to low level regions. And so that gives you a way to kind of like watch recurrency in action, by using both the temporal and spatial resolution of these combined methods, right? So, here's another example. If you want to go beyond just sensory processing, right? Cognition, of course, is much larger than that. There are much more ineffable processes at work. So here in this example, participants made judgments on tones and gabors, and they modulated those conditions with different distractors, right? There's a small, like a one back task. And this, by the way, is an fMRI and EEG rather than MEG example. So the first analysis was this familiar, spatiotemporal fusion movie, right? Here you see some frames from it. But the authors took this a step further. They used this as a base to, parcelate the response into these clusters. So clustered by the similarity of their individual time courses. And then those clusters describe several networks. And the spatiotemporal dynamics of these networks were described by sort of these four functions, which are interpreted by the authors as spatiotemporal components of attention, right? So you can see these large scale sort of stimulus independent or dependent global dynamics of attention networks that you can sort of unpack with the fusion method. So, and in the final example, I want to take it a little, in a similar direction, away from just stimulus processing to something a little more amorphous. Processing a stimulus depends on what you're supposed to do with it, right? So here the authors presented subjects with visual objects in different categories, but they cued the subjects to perform one of a couple different tasks, a perceptual or conceptual task. So at any time a stimulus was presented, there was this processing at work just by looking at it, but they had to perform, a delayed judgment on the object. So the fusion doesn't intrinsically distinguish between processes happening at the same time. So the way the authors got around it in this case was to operationalize, to model the object or task dynamics, right? The similarity structure of just objects for the task. And then they generated what's called a commonality time course. In effect, they just saw, they just determined how much of the shared variance between fMRI and MEG, so how much of the fusion time course was accounted for by object or task at a given time in a given ROI. And through this method, they saw that in some ROIs, in some parts of the brain, the overall brain dynamics cares much more about the task that's being assigned in that trial. So you can see by the blue trace, than about the specific object being represented, right, being presented here, as you can see in the red. In contrast to other visual regions that seem to care much more, at least initially, about the stimulus itself than the underlying task context. And so in this way, you can differentiate different components of sort of this overall task structure. Okay, so we've seen some of the advantages of and applications of this approach. It's highly applicable to any current questions that are currently accessible to, spatiotemporal imaging. It can be combined with other methods and representations within the RSA framework. Your RDMs can come from computational models or behavior, right? And any given method that improves the SNR, for example, of one of these components will benefit the entire, the fusion framework. It's a thing that benefits from any of its individual components being improved. And of course, it has some limitations. You don't get trial-to-trial variation. You get this sort of aggregate structure, right? And fMRI and say MEG are differentially sensitive to different parts of brain activity. And so what you get from it is just those aspects that are common where the isomorphism holds, right? Where both measurement techniques can pick up these contrasts. And by virtue of the correlation, you need stronger SNR to get a signal compared to just the individual modality analysis. Now, by way of looking forward to the potential, I'll show sort of an older figure from Nico Krieges-Korte. And it just shows that the RSA framework is vastly versatile and adaptable. So within the human neuroimaging domain here, this is really where the MEG fMRI fusion lies. You can combine it with clinical populations, or over development, or across task manipulations, or correlating brain signals to computational models, which of course is a hot topic these days. So, of course, everything that I've said here, I've learned in the course of collaborating with my colleagues, most of whom are shown here. And here is a reading list, some good current and upcoming reading for most of the work that I've cited here. All right, well, thanks very much.